So just out of curiosity, how many of you went to mentoring through today? You? Not many? Okay. Well, you made it through your first full week. First week of lab.
Lab went very, very well.
Lab went well, good. In response to some questions we've gotten, your lab quiz, your pre-lab quiz will always be at the beginning of your lab section, and you can always have notes with that. Again, the intent is to get you to leave the lab before you come to lab. On Monday, we will be talking about the upcoming lab. There's probably not a whole lot we'll have to say, although we could give them some kind of background about use of the microscope and stuff, I guess, in that session.
Easier when you have one in front.
I think that was it. Oh, in there. Okay. Well, we'll do a little bit. Go behind the chair. Your first textbook reading quiz will be at the end of this section, at the end of the period on Monday. It's a 10-point quiz. Actually, there are 11 questions. You could theoretically get 11 out of 10. It's kind of a bonus question on it. And again, it's intended to determine whether you have been reading the textbook or not. You have a problem with your writing implements, aren't you?
Everything's photo.
So, bold-faced terms are good to write down. And as I said, don't forget to look at those chapter summaries that the author has done for you at the end of the chapter, because he's basically summarized the chapter. And then the self-quiz questions at the end, too. It's not uncommon for some of those self-quiz questions to actually be on the quiz. We'll also have time on Monday to do a little bit of wrap-up of last week's lab. Hi, come in.
Sorry.
Welcome. Your lab hand-ins for last week's lab will be due at the beginning of the lab period. But in our Monday session, if there are any last-minute questions that you need to have answered, make sure you ask those then. So we'll use Monday to kind of wrap up a previous week's lab and then to introduce the next week's lab. Okay? Anything else? So, what's an antibody? We've all heard of antibodies before, right? In what context? Pardon me?
Immune system.
Immune system, okay, so they're a component of the immune system. But what actually are they? Some kind of a substance, aren't they? And specifically, they are a biological molecule. You know what class of biological molecules they belong to? They're proteins. So if they're proteins, where are they going to be synthesized? On ribosomes? Okay. How is a cell going to know how to make that particular antibody? Is there one question? Is there more than one type of antibody? Yeah, there are, millions of different types of antibodies. What do they do? Why are they part of the immune system?
Recognize bacteria or infections.
Okay, they recognize other molecules which can be part of bacteria or virus or other infectious agents. And those molecules that they recognize should be foreign to the body. Why is that ability to recognize foreign things as opposed to self molecules important? What if you have antibodies that start reacting to your self molecules? Is that good? No. In fact, it's a type of disease. What do they call it, disease? That's an autoimmune disease, and there are a lot of different types of autoimmune disease. these foreign molecules that the antibodies react with are called antigens. So antibodies react with antigens in a very specific way. We're going to be dealing with in a few weeks, both in lecture and lab, another very specific protein-molecule interaction. We're going to talk about enzymes. And enzymes bind very specifically to particular substrates and catalyze a reaction. Well, there's a similar type of specificity between antibodies and foreign antigens. And what does that interaction result in? What happens when an antibody interacts with an antigen? Is it good for the antigen? No. It ultimately results in elimination of the antigen. Okay? So there's a whole bunch of different kinds of foreign molecules out there in the world that are threats to your body and cells. And so your body's immune system has the ability to make all these different antibodies to respond to these different foreign antigens. How does it know how to do that? Because every different antibody has a different primary structure, a different sequence of amino acids. So how does it know to make antibody A versus antibody B? Or antibody C or D? It's got to have instructions, doesn't it? And they're encoded into DNA molecules. So there are antibody genes. Okay? Should your cells produce these antibodies if the antigen isn't around? No, it would be a waste of resources. Okay? You'd be making something you don't need. So at some point in time, if you're exposed to a foreign antigen, there is a signal that is sent to particular types of immune cells that say, start producing your antibody. And basically, those antibody genes become activated or turned on. And then the cell eventually starts producing antibody. And what does it do with the antibody it produces? Does it stay inside the cell where it's made? No, it's going to be secreted, right? Into the body fluids. The blood or the lymphatic fluid. And once it's secreted, hopefully it will find the foreign antigen that it's paired with and result in its elimination. Does anybody know the names of the kind of cells that produce antibodies? Technical question probably at this point in time. The specific cells that actually produce the antibodies are a type of cell called a B lymphocyte. So B lymphocytes have to be activated. Their genes are turned on, and eventually that antibody is produced and secreted. There's another term for turning on a gene. Another way to state that is to say that the gene becomes expressed. It was off, and now it's turned on. That's gene expression. What does gene expression involve? DNA being transcribed into RNA, and RNA being translated into the antibody protein. So where does gene expression begin and what cell organelle? Where does this process of transcription take place? Remember? In the nucleus, okay? So genes become activated. The gene is copied into a messenger RNA. And then what happens to that messenger RNA that encodes the antibody structure? It has to leave the nucleus, go out into the cytoplasm, associate with a ribosome. That's translation. And because this is a secreted protein, what kind of ribosome is the message going to associate with? Free or membrane-bound? Membrane-bound ribosomes. Remember, they're the ones that make secreted proteins? And where are we going to find those membrane-bound proteins?
Floating around the cytoplasm.
Membrane-bound.
Oh, membrane now.
Yep. What membrane?
The rough ER.
Rough ER, rough endoplasmic reticulum. And so those particular amino acids are going to be assembled together to form a polypeptide chain. And that polypeptide chain is going to actually snake its way into the channels of the endoplasmic reticulum. That circus balloon that I said was kind of the analogy for the ER membranes. And if it's going to be secreted, where are those particular proteins going to have to end up? Outside the cell, and to get there, they're going to have to go to the cell membrane first, and then be released by what process? Remember what that process is called? Exocytosis. Okay. What are the little transport vehicles, if you like, that pinch off of the ER and travel to the membrane column? Those are vesicles. Okay, those little membrane-bound sacs. Well, as it turns out, antibodies are not just polypeptide chains. They're polypeptide chains plus some other molecules are added onto those polypeptide chains. A whole different kind of molecule. Not more protein, but instead sugar molecules are added on. Carbohydrates. So antibody molecules become modified before they're secreted. Sugars are added to them. So those vesicles that contain the original polypeptide that was made in the ER aren't going to go directly to the cell membrane first. They need to make a stop along the way. And during that stop, that modification takes place, adding these sugar molecules on the protein to make the glycoprotein antibody. So where does that happen? Where does that secreted protein get chemically modified before it continues on to be secreted? Which one? Lysosome, mitochondria, storage vacuole, or Golgi apparatus? 1, 2, 3, or 4? See a 4? See another 4? See a 2, 4. Golgi apparatus. So what does that look like? Right here a little bit. Whoops. So here's our rough endoplasmic reticulum, and all these little dots right here represent the ribosomes. And so if in this lymphocyte, if the cells receive the proper signal, those antibody-producing genes are going to be turned on. The genes are going to be expressed. That means that transcription is going to happen within the nucleus. We're going to form messages that encode the information for that antibody molecule. That's going to lead through the nuclear pores. And those messages will associate with these membrane-bound ribosomes. And then the ribosomes, with the help of transfer RNA, remember, which grabs amino acids, it's going to start hooking those amino acids together to form a polypeptide chain. And whatever sequence that polypeptide chain is will be specified by the sequence of the nucleotides in the gene. Okay? The proteins that are produced are going to start to snake their way into the channel of the endoplasmic reticulum. So these little circles right here would represent antibody molecules. But they're not finished yet. They need to be glycosylated. Sugars. Need to be added onto them. So we package up these proteins in a little vesicle. And that vesicle moves through the cytoplasm,
until it encounters this new set of membrane-bound organelles, the Golgi apparatus. The membranes of the vesicle fuse with the membranes of the Golgi apparatus, and now those proteins are released into this channel-like system. And attached onto the membranes of this Golgi apparatus are the enzymes that add the sugar molecules to the protein. It's almost like an assembly line. Okay, and as they pass through the channels of the membrane, we glycosylate the proteins. So by the time they get to the end of this, they have now been chemically modified. Okay. There are other kind of proteins that are modified in the Golgi apparatus. Sometimes lipid molecules are added to the proteins. And therefore the product is not called a glycoprotein, it's called a lipoprotein. Anybody know of any lipoproteins that are important in the body? Has anybody had your cholesterol checked recently? Have you heard the terms HDL and LDL? What do those stand for? High density lipoprotein and low density lipoprotein. Okay? So sometimes proteins have lipids added to them before they're secreted. What's the purpose of these modifications? Well, it's really hard to generalize. Sometimes the purpose of those modifications is basically to put an address tag, a molecular address tag, like a zip code, onto that protein, which will then determine where it goes next. One address tag might say, okay, you're ready to go and fuse with the membrane and be secreted. Another address tag might say, well, you got to go here next. Maybe you're going to be stored for a while before you're secreted. Or maybe you're going to accumulate actually by fusing with other vesicles into a whole new cell organelle. So some people like to use the analogy to the Golgi apparatus as almost kind of like a post office. And it adds address tags onto molecules that helps to sort where they end up. Helps to provide a destination code for them. Okay? So, in the case of our antibody molecule, once we add these sugars onto it, new vesicles pinch off of the Golgi apparatus, fuse with the plasma membrane, and now these glycoprotein antibodies have been secreted into the environment, and they might be released into the circulatory system or the lymphatic system, and then they can hopefully find their partner antigen, bind to it like an enzyme binds to a substrate very specifically, and then ultimately result in its elimination. It's a very cool system, and it is something that is incredibly important to your body, because one of the things you might have come to an appreciation for last semester when you worked with bacteria, there are bacteria everywhere, and you're constantly exposed to them. So why don't you get sick more often? Because you've got a very robust immune system, and antibodies are a very important part of that immunity. Some of the modified proteins that are packaged up in the Golgi apparatus pinch off vesicles, and several of those vesicles then move through the cytoplasm and actually fuse with one another to form a new cell structure. And the molecules that are inside of those are not antibody molecules, but instead they're enzymes that are involved in intracellular digestion. Those particular enzymes are a class of enzymes called hydrolases. And they catalyze a reaction called hydrolysis. Remember from a couple days back when we showed how polymers can be broken down? We said hydrolysis splits the bonds of a polymer
and chops off monomers.
Hydrolases include the enzyme DNase, lipase, protease, amylase. There's a whole long list of these hydrolase enzymes that can accumulate in these organelles that were formed by various vesicles fusing together, each of which contains its own digestive enzymes. What do we call this organelle? Is it a storage vacuole? Is it mitochondria or lysosome? It is a lysosome. And so again, when you're talking about kind of word association, lysosomes, you should think intracellular digestion. So, why do we need intercellular digestion? Why do we need a mechanism for having intercellular digestion? Well, there's a phenomena that's really important in some types of cells called phagocytosis. That word literally means cell eating. It's a way for cells to consume other cells. One type of phagocytic cell that's really important is a type of white blood cell called a neutrophil. And you've seen neutrophils in the inner life of the cell. That neutrophil that was moving through the circulatory system and eventually made its way out of the vessel into the surrounding tissue was a white blood cell and probably a neutrophil. And what they do is they gobble up other cells or pieces of junk or potential food. And they do that by pinching in their cell membrane and trapping what was ever in that vicinity inside of that little sac and bring it into the cytoplasm of the cell. That little sac is called a vacuole. So this might be some bacteria, or it might be other things that are potential food resources. Well, what is food? It's really organic molecules. I mean, cells are made up of protein, carbohydrate, lipid, nucleic acid. Okay? But can the cell, in its metabolism, like cellular respiration, use those molecules in the polymer form? No. What they need to do is to chop it up into smaller bits or pieces and to break the polymer into a monomer. How do they do that? They use these particular enzymes. So what's going to happen? That food vacuole is going to fuse in the cytoplasm with a lysosome. And the membranes of those two cell organelles are going to merge, and now everything is going to become mixed together. And now these particular food molecules or bacterial cells or whatever it is are going to be exposed to these degradative enzymes, and it's going to be chopped up. And then once you have amino acids produced or fatty acids, those can then move out of the vacuole and into the cytoplasm and be used in cellular metabolism. So this whole idea of intracellular digestion is really important. These particular enzymes are referred to as acid hydrolases. The reason that we specify that is that it's actually quite acidic inside of the lysosome. And these particular enzymes have a pH optimum that is acidic in nature. That's going to be significant. We're going to hold on to that for just a second. Talk about that later. Another thing that can be done with these lysosomes is that the lysosome can fuse with an old, worn-out cell organelle, like a mitochondria. And in the process, the old, worn-out mitochondria can be broken down and basically recycled. Because what is a mitochondria? It's proteins, carbohydrates, lipids, nucleic acids. So we take this old, beat-up mitochondria that doesn't work anymore, and we break it back down into its components, and then we can use those to do what? To make new cell parts, or to provide energy for the cell, for its function. That particular process is called autophagy. Literally means self-eating. The cell's kind of chewing up part of itself. Sometimes cells will literally kill themselves using this particular process. Lysosomes start fusing with other components of the cell, and the cell literally starts self-destructing. When a cell purposely self-destructs, that particular process is called apoptosis. Can you think of an instance where a cell self-destructing might be desirable?
If it's not ready during cell division, like the stages, if it gets checked and it's...
Very good, yeah. If it hasn't gone through the proper checkpoints in cell division, and therefore it might be abnormal, we could then go through apoptosis. What if a cell becomes cancerous? Might we want to get rid of it before it can start dividing? Actually, programmed cell destruction is also important in some phases of development of particular types of organisms. What's the difference between, or one of the differences between a frog and a tadpole? Tadpoles have tails, right? Frogs don't. Why does that tail get lost? Because of a programmed cell destruction that's part of the developmental process that results in the destruction of that particular tissues. We all start out as embryos without space between our digits. And eventually that webbed hand, fingers start to separate. There's programmed cell destruction that results in the breakdown of some of the tissues, so we then have separate fingers. So it's very important to the developmental process as well. True or false? Malfunctions within a lysosome can cause diseases. True. These particular diseases are called lysosomal storage diseases. There's a whole long list of them. It might be because one or more of the enzymes within the lysosome is defective. It might be because the lysosome can't fuse properly with other vacuoles, for example. Typically, as a result of these diseases, some bad things start accumulating in the cells that should have been broken down. There is one lysosomal storage disease that has been very well studied called Tay-Sachs disease. Have you ever heard of that before? Tay-Sachs disease is the result of an accumulation of certain types of lipids in the cell. And that can cause neurological problems. So there's a whole long list of these lysosomal storage diseases that have been discovered. Okay. Another cell organelle that is part of the endomembrane system are vacuoles. Which of those statements about vacuoles is correct? We already talked about this one, right? We said a food vacuole can be formed by the membrane pinching in. That's endocytosis, and bringing stuff into the cytoplasm. They may store substances the cell will need later. That's called a storage vacuole. They can be formed by smaller vacuoles or vesicles fusing together. And in plant cells, they're oftentimes filled with water. In plant cells, the cytoplasm of the cell is dominated by a big, large central vacuole that really does take up a lot of the space of the cytoplasm. Here's a good example of a type of vacuole that's important in paramecium in this particular case. This is a developing water vacuole. Water vacuoles are formed by little small vacuoles surrounding molecules of water and infusing together to form a big sac of water. Here we've got a vacuole that has become filled, and then it fuses with the cell membrane and pumps the water out of the cell. Why is it really critical that this vacuole system works in a paramecium? What would be the consequences if it didn't work properly? This is pumping water out of the cell. What would happen if we didn't do that?
The cell in theory would drown.
And even worse, as it fills up with water and fills up with water, what's going to happen to that cell? It's going to burst. It's going to lyse. Water is constantly rushing into these particular cells because of their external environment, and specifically because of osmosis. We'll have a lab about osmosis the week after next. But these particular type of cells are living in hypotonic environments. And in a hypotonic environment, the water enters the cell, and the cell swells and develops an internal pressure called turgor pressure. And eventually that cell could burst. So what do? We pump the water back out again. Okay? Kind of like living in a leaky boat. As long as you can bail out the water faster than it's leaking in, you can maintain an equilibrium. You can still float. You can do an interesting experiment with paramecium where you can expose them to a drug that actually interferes with these vacuoles forming and discharging the water. You can actually see the cells swell and swell and swell and then pop and burst. So contractile vacuoles are really important to a lot of aquatic organisms. When we talk about plant cell structures, we're going to come back to vacuoles and talk about the central vacuole in plant cell, and also talk about storage vacuoles in plant cells, because they have some interesting implications and some interesting functions. So, which cell organelle is a structure adapted for making ATP? Mitochondria, right? Sometimes called the powerhouse of the cell. What cellular metabolic process is going to be taking place in the mitochondria? What makes ATP? Cellular respiration. Now, as it turns out, cellular respiration has various parts. One part, glycolysis, happens in the cytoplasm of the cell. But then in aerobic cellular respiration, which involves oxygen, the Krebs cycle and the electron transport system take place within the mitochondria. So, here's a representation of a mitochondria. Little kind of sausage-shaped organelle, which, as I said earlier, has ribosomes and DNA. This suggests that the origin of the mitochondria is
bacterial in nature, because the ribosomes are 70S variety, like prokaryotic ribosomes. Also, the mitochondrial DNA has characteristics more of prokaryotic DNA than eukaryotic DNA. Speaking of diseases, we're also finding out now that there are a lot of diseases that are associated with defective mitochondria and defective mitochondrial DNA. I love the mitochondria. The mitochondria is an organelle whose structure is just very elegantly adapted to its function. Its function being producing ATP. When we talk about cell respiration, one of the things I will constantly do is I will use this classroom as kind of a model for mitochondria. Mitochondria has a very interesting organization, and the key to that is the fact that it's got two membranes. It's got an outer membrane and an inner membrane. So, in that particular model, what's this room going to be? If you walk out the door out there, you're in the cytoplasm. These walls represent the outer membrane of the mitochondria. This one right here. This group of chairs, the outer group of chairs, is going to represent the inner membrane. And one of the things that you're going to see is that that inner membrane is highly folded. What is that an adaptation for? Increasing surface area. Because the more surface area you have, the more efficient you can be in making ATP. This space between the walls and this outer layer of chairs is right here. It's called the inner membrane space. Everybody else's chairs that's in the middle of the room represent the matrix of the mitochondria. One of the things that I will stress over and over again is that in order to make ATP efficiently, you need to have a membrane, the inner membrane, separating two spaces. The inner membrane space here and the matrix. We'll talk about why that's the case when we talk about cell respiration. But the organization of the mitochondria is crucial to be able to make ATP in the most efficient manner possible. Okay, good place to stop. So if you do not have to retake mastery quiz, you're good to go. We'll see you on Monday. And if you do need to retake mastery quiz, it's up front here. Come up and grab a copy and go to it.
I can grab one. Does it matter?
Forgot the seating chart again.